Hi, for this recorded video, we will be discussing the pre-lab for experiment 8 titled Spectrophotometric Determination of Iron 2 in aqueous solutions as a complex of 110 phenanthroline. So this recording as well as the slides presented in this lecture discussion are protected by the university's copyright policy and the Intellectual Property Code of the Philippines. So the objectives of this experiment, of course, is to review your principal knowledge, so the concepts of UVVs, spectrophotometry. So it's expected that you have some sort already of background knowledge from your Chem 17.1. Okay? So you should also know the proper instrumentation and the operation of the double beam UV-Vis spectrophotometer. So in your Chem 17.1 classes, so you were taught how to use a single beam UV-Vis spectrophotometer. So the operation of the instruments are almost similar. Okay? And of course, the last objective of this experiment is for you to know the use of the UV-Vis spectrophotometer for the quantitative analysis of inorganic ions or compacts. Okay? So we will be applying here your knowledge on the Beer-Lambert's law. So let's take a look at the experiment. So in this experiment, you will be dealing with the iron 2-110 phenanthroline system. Okay? So your analyte here would be the iron present in your sample, okay, in the form of iron 2 plus. So the divalent form of iron. Okay? In particular, you will react this iron 2 plus with a ligand called 110 phenanthroline. Okay? So the reaction of iron 2 with 3 moles of this ligand will form an octahedral complex, so called tris 110 phenanthroline iron 2 complex. Okay, so this is a colored complex. And since it is colored, you can use UV-Vis spectrophotometry to determine its absorption properties. So you can obtain its spectra, absorption spectra, and determine its wavelength of maximum absorbance. And of course, you can measure the absorbances of the solutions. Okay? And if you have a sample solution containing an unknown amount of iron in the form of iron 2 plus, so you can determine the concentration. Okay? So iron 2 plus reacts with 3 moles of 110 phenanthroline. So it's also called as orthophenanthroline, okay, to form your complex. Okay? So in this complex, you have your iron 2 plus as the metal center, and you have your 3 phenanthroline ligand. So 110 phenanthroline is a bidentate ligand. So meaning that your metal attaches or binds to it at 2 sites. So which are the electrons? So the lone pair of electrons from your nitrogen atoms. So from the nitrogen atoms of the phenanthroline molecule. Okay? So the complex formed from the reaction of these two forms a red-orange, or sometimes it's a very intense red complex solution. Okay? And again, since you have a colored solution, you can measure its absorbance using UV vis. Okay? So for this experiment, you need to prepare the following solutions. So you'll prepare 6 calibration standard solutions. Okay? So you will add appropriate amounts of your working standard iron 3 solution. Okay? So take note that although we are measuring iron 2 plus as our analyte, and here in the experiment, so we are preparing your solution from iron 3 plus. Okay? So you need to take into consideration here how will you convert this iron 3 plus to iron 2 plus. Okay? So you will add these amounts of your working standard iron 3 solution into your volumetric flask. Then you will add these two reagents. So 1 ml of 10% hydroxylamine hydrochloride solution. Okay? And then from that, so you can now add your ligand solution. So around 5 ml of 0.20% 110 phenanthroline. And before you dilute it, so you will add a buffer. So around 1 ml of an acetic acid buffer, pH 4. Okay, before you dilute it to Mark 2 using distilled water. Okay, in particular, so let me just point out here. So you need to use a special kind of distilled water called distilled deionized water. Okay? So if it is available in the laboratory, so this you should use this instead of the usual distilled water for the UVVs analysis. So otherwise, so the distilled water would do fine as well. Okay. So after adding all of the reagents and diluting the solutions to Mark, so mix it thoroughly. Okay. And you could now use this in the analysis, so in the preparation of your calibration curves later in the experiment. Okay. So for this experiment, so you will be using the double beam UV-Vis spectrophotometer. So you should know how to prepare the instrument. Okay, so your instructor will be the one to prepare the instrument. Okay? But you should know how to load your sample and standard solutions using the cuvette. So using the cuvette into the UV-Vis instrument. Okay? And then you will determine the analytical wavelength. So this is your lambda max of your sample. So using the spectrum mode of the UV-Vis spectrophotometer. Then after that, so once you've determined the lambda max of your um, sample, so you could now set it into your instrument and you can now determine and generate your calibration curve. So by measuring the absorbances of your prepared calibration standard. So you can do that by using the photometric or the quantitative mode of the UV-Vis instrument. And after you've already generated your calibration curve, so you could now use that particular calibration curve to determine the amount of your analyte in your sample. So you'll just measure the absorbance of your sample solution. So at that particular wavelength as well. Okay? So at the lambda max. And record the absorbance reading and then plug it into the equation of the best fit line that you've obtained from your generated calibration curve. Okay? So please familiarize yourselves with the steps on how to operate the double beam UV-Vis spectrophotometer. So you could find that in the appendix C of the Chem 28.1 lab manual. So that's it for the pre-lab for this experiment.